even pairwise. Single sequence.
Welcome to the AI Biological Science Hackathon! The Nobel Prize in Chemistry was just won by AlphaFold. We are at a moment in historical progress where there's a massive overhang of AI technology which can be applied to biological research. And the results that have been coming out of so many of the speakers that you'll hear from today are phenomenal. It is a joy to be with you on Saturday morning to hopefully co-invent some of the future of this glorious frontier in biological research. My name is Jeremy. I'm the founder of AGI House, so welcome to AGI House. The intention here is really to bring the creators of fantastic technologies to co-build the future of artificial intelligence technology, but really to envision in a really creative environment projects that cross the boundaries of companies and of fields. In this case, we're crossing the fields of artificial intelligence and biological research, and come up with projects that can be implemented in a short period and launched that will take advantage of the phenomenal technology that we have all been handed. It's just a wonderful moment to be alive with glorious view and a fantastic suite of people. The conversations we've had here have been great. So a quick preview of the evening. Sorry, this is basically we have a speaker set. So we'll have talks from 11 founders, mostly of fantastic AI cross-bio companies, and these talks are going to be pretty inspirational. I heard a quick preview of what Andrew will be speaking about. I'll talk about research generation, but basically a lot of the projects that we build can be inspired by the founders who are in this room. We all have a chance to meet and interact with. If you are interested, sign up for a lightning talk. What is a lightning talk? A lightning talk is a brief 5-minute description of a project that you'd like to build today, and it'll let you get other folk in the room involved in your eventual presentation. So the plan overall is we're going to have some talks. The hackers are going to present their ideas, and then we'll discuss forming teams. We'll basically be building for 7 hours, and then at 8 o'clock, so in just 7.5 short hours, everyone will present the projects that we've built today. And personally, there are a lot of papers that have inspired me. I'm very inspired by the work on diffdoc at MIT, where the docking problem, if solved, can sort of dramatically improve our ability to control, proteins and generally, there's the corollary to AlphaFold, where you're interested in the protein folding problem, but you want to expand it to a number of other domains. Evo is out, and they have a really clean API. Future House has put out some research literature review automation technology. There's just a tremendous amount of sort of raw material for worthwhile projects, and you'll hear presentations of potential projects as well. So all that said, any questions about how things will run? It'll be about 5 minutes per talk, so if you're speaking, do devote 4 minutes to your presentation and then at least 1 minute to basically how artificial intelligence can be worked into a practical project where you have some insight into how your company or organization's been dealing with all of these new technologies. So that people who are making decisions about what to create can potentially do something that would advance your conception of how the future of AI and bio should play out. And with that, let's hear it for Andrew White from Future House.
So just open up a new tab. Hey guys, I'm Andrew White. I'm from Future House. Future House is a non-profit based in Dogpatch, right here, San Francisco. And I have a couple slides that I worked really hard on, so I'm going to show you guys. So we'll look at these. And honestly, I just have 4 slides. Okay? So this is really cool, so we'll just look at this. Wow, thank you. So Future House, we're a nonprofit. We're funded primarily by Eric Schmidt, but we have backing from other organizations and individuals. Based in San Francisco, 18 employees. And we have a wet lab. And that's a wet lab. You guys can come check it out. So science is changing independent of AI. This is the number of papers basically submitted to archive per year, and you can see ChatGPT was like not even, you know, it's been a trend that's been going on. AI is, or science is becoming harder and harder to do. And if you look at the complexity of science, every year there's more authors on every paper. So, you know, whereas 30 years ago the average paper was like a single author thing, it could all fit in one person's head, we're getting to the point where papers require like 5 people. And as we get to like the next stage of biology and AI, we need AI just to sort of rescue productivity in biology. The next challenges are things like building a mechanistic model of the brain, annotating all proteins of unknown function in the genome, and then understanding, you know, the gene regulatory network. So these challenges are just, they're big. They won't fit in one paper, they won't fit in one set of equations. And so I think we need AI as like an actual tool to challenge, to do these very challenging tasks. So the mission of Future House is basically automating scientific discovery. So we're building agents to do this, and we're building environments to test them on. And so what I was really hoping to present today was our new language agent framework. So we're going to be open sourcing at the end of the month, hopefully, a language agent training framework. So think like PyTorch for compute graphs of agents. And we're going to release a gym of environments that we think are meaningful for science. I'm going to hold back the identity of the environments in the gym because I think it'll be fun. But I will tell you, many of them are related to biology. The things that you think are important in biology will be there, and there will be things like deep learning models will be part of the tool set that the agents use. So I think it's going to be really exciting. But today I'm just going to talk about my hackathon. So I actually did the hackathon last night because I have 2 small kids, so I do my work at night instead of during the day. So I put out a little blog post about it. So what I did is that I just wanted to get like this, I've been really impressed with these very long context models like Gemini. So, and I was using our tools. We have this thing called Paper QA, which does like, it writes like literature reviews, as Jeremy mentioned, but it also aggregates clients from a whole bunch of different like metadata providers about research papers. So one of the clients is something called OpenAlex, which basically has PDF links for every scientific article if they exist. So it goes across the internet to find PDF links to articles. So I downloaded 514 PDFs about crows. So I found a bunch of research papers about crows. I downloaded all of them, and then I put them through Gemini. And I had a little guide that I wrote. So me and Gemini worked together on making a guide on how to make a good factoid for social media. Then I put this factoid into all of the papers, or sorry, I put the factoid guide, put all the papers into the LLM, and then I had it generate a bunch of crow facts. So from 514 PDFs, I was able to get to 692 crow facts, and then you can now go here and see all the crow facts. And I know that this is like a hackathon, so I have made an API. So if you go to facts.drugcrow.ai, it will deliver a JSON of a crow fact. And like our whole thing about writing agents for doing scientific research is that they all give you the citation of where the fact came from, and then it gives you a direct quote from the paper about how it knows that that fact is real. So here's the p-value for this fact. So cool. I think that's all I wanted to say. I'll just mention that on my blog post I have another sort of one-day project that we did, which was estimating the cost of every single scientific discovery in the history of human civilization, which is another kind of like fun little project here. So like here's the cost of inventing, geometry by building the pyramids. Here's the Higgs boson discovery by building CERN. Anyway, it was a fun little exercise where instead of using papers, I took all the Wikipedia articles for every scientific discovery on Wikipedia and had O1 do a little like Fermi kind of cost estimate, seeing how many people worked on it, how many hours, what the capital costs. And so this data is also available in case somebody wants to look at something like, how are discoveries changing over time, anything there, that's available here. So, great, have a great time. Super excited to see what you guys build. Thank you.
Thank you, Andrew.
You're wearing a crow shirt as well.
From E11, we have Andrew Payne.
No.
All right. Hi, everyone. I'm Andrew Payne, co-founder and CEO at E11 Bio. Thank you to the organizers for inviting me, and let's talk about the brain. So, show of hands, who saw the fly connectome drop 2 weeks ago? Yeah. Who knows what I mean when I say connectome? Okay, not bad. Okay, the wiring diagram of the brain, the graph. We now have, for our favorite model organism, Drosophila, a graph of 130,000 nodes and 50 million edges that basically describes how its brain is wired up. And this is transforming neuroscience. We are now looking, I don't have permission to play this file. Sorry, hold on.
Can I download it in Network X format? I really want to download it.
Look at that, amazing. Okay, so what I was saying was that it's transforming neuroscience. We are now able to simulate the entire dynamics of the brain of a fly using ML constrained by real graph connectivity in the brain. So this is moving super fast. I think we're going to see simulations of how the fly thinks a lot sooner than most people expect. But what I want to focus on today is actually what comes after the fly. And by the way, if you haven't looked at the special issue of Nature from like 2 weeks ago, check it out. It's super cool, goes into great detail, amazing work. But what comes next is how do we go from something like Drosophila to the mouse? And there are good reasons for doing that because one, we have no idea how connectivity changes in brain disorders. And if we can figure that out, that's gonna unlock new opportunities for like therapeutics and BCI. And two, the brain is still our best example, existence proof of like general intelligence. And if we can just glean a couple of insights from the brain for how to design the next generation of AI agents that can think over multiple steps or are more energy efficient or all of the different things that we can do, that would be huge. So that's why we want to go after the mouse brain. But it's 1,000 times bigger. And so there's a bottleneck. And the bottleneck is looking at images of the brain, petabytes of these images, and segmenting them. Most people here, who knows what segmentation is for computer vision? Yeah, you're discriminating between objects in an image. And we can do this really well, but at the kind of scale that we're talking about, current performance is not good enough. And so the way that we deal with this is that we have human annotators go in after you've done an automatic segmentation and clean it up. They paint pixels manually because the humans have learned how to discriminate what cells look like way better than the current models. And this works, and this is how we got the fly connected. And it only took 33 person years of painting pixels to make it happen. So if we're going to do something 1,000x bigger, we need more performance here. And so this step, proofreading, is the real bottleneck. It's like 95% of the cost of brain mapping. If we were going to scale up what we have today, it would be billions of dollars to do a mouse brain. So this is the point of leverage for all of you. If we can solve this problem, then we are looking at real maps of mammalian brains. And shameless plug, just a couple of years ago, some friends and I got together and we started E11 Bio, which is a nonprofit focused research organization in the Bay Area, building tools that will catalyze next generation connectomics. And we are attacking this problem in 2 different ways. One is that we are using synthetic biology to engineer the brain cells of the mouse to proofread themselves. We deliver something called prism tags to the brain, and that makes each neuron a different color using a protein-based barcode that can be discriminated in images. And then the second thing that we're doing is we're building new model architectures that can take advantage of this prism information, this color information, in order to get segmentations that are 100x faster and cheaper than and more performant than what we have today. And so when you put these 2 things together, we can go from black and white images of the brain, which is what people use right now, to color images that can be easily discriminated, to low-cost segmentations of entire brain areas. This is a sneak peek of our first big data set. This is the CA3 region of the hippocampus. We're doing a big data drop sometime next year, and we're pretty excited about what this means for us, because at this point, we've already mapped something about the size of the fly brain itself, and we're just getting started. So, in summary, one, if you care about brain medicine, or you care about brain-inspired AI, we got to get the mammalian connectome. We need to solve proofreading to do it. Genetic barcodes give us a pretty good angle of attack, but it's not a magic bullet. We still need better models. We still need better tools to actually make this problem affordable and tractable. So here's how you can get involved. First, just check out the fly connectome. It's super cool. You can do it today. You can hack on it today. It's right there. Second, if you care about brain science and want to learn more about this area, you want to see like whether you have an angle of attack, sign up for our mailing list. We have events, we push updates, we have sometimes hackathons and early access to data. So, hang out. And also, we are hiring. We love people who are talented and passionate and want to work on something that could change the world. So, maybe you want to join us. We'll see. Thank you, everyone.
Can you post a link to the Connectome direct download in the Slack?
Yeah, I actually already posted that. Yes. But I think hackathon projects that involve that.
Yeah, just look at it. It's so cool.
Okay, that was Andrew Payne. Next we have Atray, Dixit.
Atray.
I sent you a PDF if it doesn't work out.
Hopefully it's good.
Just do it.
Whatever.
Oh, you don't know. We have one flight. Well, we have two, I'll let you know. The section up to the next. Yeah.
There we go.
I need that graph.
Sweet. Can you guys hear me?
Yeah.
My name's Atray, founder and CEO of Onco, new company. We haven't finished our seed round yet, so I actually won't talk tons about the company. I'll just give a quick intro on what we're up to, and then focus on some challenges that are related to what we're building. And if one of you guys can solve, we'd probably pay you for it as soon as we have money in our bank. Onco, the name is oncology and then the binomial coefficient n choose k. So we're trying to work on combination therapies in cancer. That problem is a big problem because we have 20,000 genes, which means if you want to look for 2A drug combinations, it's 20,000 choose 2, which means it's 200 million, and it just kind of goes exponentially from there. Have a really cool team, won't dwell on it, but myself, I've been working on data generation techniques for target discovery, drug discovery for a while, and this is one of the exemplars in cancer. So between 1940 and 1988, we went from 0% cure rate for pediatric ALL to 90+% cure, and it just was a sequential kind of addition of therapies to this cocktail. And this is just doctors being smart over 50 years. So it's really cool, but we haven't done this across cancers. So yeah, let me just kind of share an interesting problem I've been thinking a lot about. So my PhD work, Perturb-seq, was about kind of generating cellular perturbation response, how cells respond to drugs or target perturbations. Cells are small, and there's public data sets of about 10 to the 5 perturbations out there. It's really cool. Companies I was talking to Natalie about Zara, and they raised $1 billion, and they're going to generate a shit ton of this data, about 6 of these per quarter, and they think that's going to be useful for ideally predicting A, which is human clinical trial outcome results. And I think most people in the drug discovery funnel think about C to B to A. We'll do C, and then we'll test it in B, and then we'll finally test it in A. And I think that's a reasonable thing to do, but I've been thinking about this / trying to play around with data sets in this space. So take all the publicly available clinical trials data, take all the preclinical mouse data out there, take all the Perturb-seq data out there, and ask the question, how well can you predict A given B? How well can you predict A given C? And how well can you predict A given B and C? And frankly, it's not very good in my attempt, and I'm not the best machine learning person out there, but let's just say the best I've gotten is AUC 0.6, 0.7, and Google released this LLM for Therapeutics thing that is in the 0.5 to 0.6 range for predicting trial outcomes. So I think that's a really hard problem, but there's a lot of public data and a lot of smart people. So I think people should be thinking about not just A or B or C, but how do you map between the three? I think it's really cool. And then this is kind of this cool like scaling law time thing that I think is interesting. So this is like perturbed seq data set generation over time, that cell level response. This is how many clinical trials have ever been done over time. And that's ChatGPT eating all the data. So, you know, it's a y-axis is a log scale. Just pointing that up. ESM, which is like a sequence function predictor that eats protein sequences. You know, that has a lot of data and is pretty cool. For Onco, where we're trying to see if we can operate in this regime. But I think like, man, how do you build a foundation model out of this stuff? I'm not sure. The other prompt that I think is really interesting that I've been trying to milk the hell out of like clinical trial outcome data, but that's just binary, like did the drug pass trials or not? And I think there's some interesting results that are hard to parse but live within clinicaltrials.gov. Like all the trials have, a good chunk of them have results posted, and that contains quantitative data about how well the drug did, which is not just binary. And I think, frankly, like folks like these, open targets, like have parsed a lot of this, but they basically just report binary data on like did the drug pass or fail. So I think if someone's like talented and can kind of extract quantitative information out of that, that would be pretty cool. So if you're interested in either of those projects, I think, yeah, as I said, at the point we have money, I pay you to do it. So reach out. Or co-founder, maybe. Cool.
Great.
What's foundation models? So up next we have Amit Deshwar, who is building stuff. Amit. Some secret knowledge about to be drawn.
So I'm Amit. I'm co-founder of a company called Kernel Labs. We build foundation models of RNA to develop better mRNA drugs. We're also fairly early, so I'm not going to spend too much time talking about what we're doing. But what I do want to talk about is 4 figures that I think people who are working at the intersection of AI and drug discovery should understand. The first is the problem. So this is the amount of R&D dollars per approved drug over time. And what you can see is that despite all the advances in biological techniques, in compute, in sequencing, we're managing to get worse and worse at developing new drugs. That it costs about 2 orders of magnitude more dollars in R&D to get one approved drug now than it did in the past. And it looks like maybe it's leveled off since the 2010s, but we're leveling it off at 100 times worse than it used to be. So this is, I think, the fundamental problem, that we're just not very good at developing and finding new drugs anymore. The second is, where are these expenses? So this is looking at how much it costs to develop a drug at the different stages of drug development. And what you can see is at the very beginning, you spend almost no money, $1 million. And as you move along the path, the costs just keep jumping up and up. So to do your phase 1 trial, you know, it depends on the drug and stuff, but say 20, 30 million, 60, 200 million. And so where you're trying to improve things controls a lot about how much value you're actually generating. That if you're just making hit identification faster, then you're solving a problem that right now costs $1 million to do. When maybe if you could improve something here, you're improving a step that is a $20 million cost. The third is the consequences of the time value of money, that the revenue that you get from developing a drug is pushed very far into the future, which is worth less than money now. And each stage has a very high chance of your program failing. And so what it's plotted here in blue is the risk-adjusted net present value of a drug as it moves across the different stages. So target to hit, here. I think this is like 1, maybe less. And it stays extremely low until you start getting into your phase 2 and phase 3 trials, where suddenly the value of your drug jumps from single-digit millions to billions. Depends, of course, on a lot of properties about your drug, but this is generally the value of the drug that you're developing over time. And then when you combine those things, where are the costs, where are the risks, when do your revenue flows start coming? This looks at each stage, what if you made it faster, higher quality, or cheaper? And what would the economic consequences of that be? And what you can see is that by far, the single biggest thing you could do is to reduce the failure rate in the phase 2 trials. And that's where you start to see, does your drug have an effect on the disease? And there are some other problems of like medium value, but this is where by far the biggest concentration of it is. And again, a lot of people are spending a lot of time hit discovery, optimization, and yet when you do this analysis, none of those things are actually worth that much. And so thinking hard about how can I go after the high value stages is really important when you're trying to pick which problem to work on. And then finally, fifth bonus thing, no such thing as an unconditional probability. All these things are aggregates across many different diseases, drug types, and what you're working on might be different. Finally, I just want to plug Krishna Labs. We are hiring, particularly on the ML side. We're building better mRNA drugs using these foundation models of RNA. I'm working with the ex-CSO of Moderna. She built Moderna's platform and wants to do it again, and yeah, looking to add to our computational team. So come talk to me if you want to work on foundation models of RNA.
Awesome.
Fantastic.
Question. Ask now.
I was just curious, like, what do you think, like, I feel like people would pitch, like, if you're 10 times better at discovery, maybe you'd have 10 times, way better at phase 2 success?
Who pitched that?
Yeah, I don't know. Well, so the problem...
Some anonymous sources.
So, okay, what do you get after you run your model and you find a small molecule that binds to a protein? You get a molecule that binds to the protein. And why would your prior probability be any higher that you're going to be able to manufacture it, that's going to distribute safely, or distribute to the right places in the body, that it's going to be safe, just because you have a great model of predicting small molecule protein interaction? And like maybe you can, but all those other processes that control safety, biodistribution, metabolism are different than what's driving the binding to your target. And so I don't actually think that's true. I think that you can have this amazing prediction of small molecule binding to a protein, and it works great, high affinity, but there's no reason to think that your probabilities of passing the other hurdles are going to be any higher.
Can I ask a follow-up?
Sure.
So the failure on phase 2, obviously many causes, but do you think it's because they weren't sure at the beginning if the target is purely disease modifying, or it's PKB, PD, like you said, it's getting whatever the drug is to the point that it needs to be?
Well, often you do get at least some PK, at least you get like PK in your phase 1. And so I think the biggest driving of failures in phase 2 is target risk. And then just like the magnitude of modulation that you're able to achieve in the clinic. That maybe it's a good target if you can eliminate 100% of the activity of your target. But when you figure out how much you can dose in your phase 1 and you put that maximum amount in your phase 2, then it's not enough modulation of the target to have the effect that you want. So it is a mixture of like target risk, PD effects, and then of course, there's like miscellaneous other things, maybe some rare safety thing comes up. There's a lot of things that drive phase 2 failures. It's not purely target risk.
Deshwar. We have a fantastic Alessandro Migliara, a research scientist at UCSF. Let's hear it for Alessandro.
Fairly quickly, can someone open the front door so we get an airflow going? Thank you.
Hi guys, nice to meet you. I'm Alessandro. I'm a senior postdoc in LimLab at UCSF, where we are designing new synthetic cells for cancer immunotherapy and regenerative medicine applications. So what we are doing basically is we are used to think about drugs as small molecules, something which is not alive. Actually, with gene and cell therapy, we're starting to think about living cells or DNA or RNA molecules can be per se drugs. We, of course, all of us are known about scientific success of this. We think about COVID mRNA as a vaccine, diabetes. We can now try to cure or, let's say, prevent it by using, let's say, engineered the proteins that are produced in synthetic cells. And of course, also, we are very well about the potential capacity of using cells per se to cure and eradicate cancer, such as this approved CAR T-cell therapy for B-cell acute lymphoblastic leukemia. But even if there are these very, very well-known success stories, actually in most of the cases when people try to develop new gene and cell therapies, they fall in this, which can fully considered as the death valley of gene and cell therapy. This is because there are different kind of problems that I want to try to focus your attention on, because actually it's where we see the potential of AI to improve, let's say, these kind of things. So there are different kind of reasons why, as those people before said, the gene and cell therapy application can fail, even before reaching clinical trials. One is the biomanufacturing scalability problem. Let's say manufacturing cells or gene is not as easy as manufacturing small molecules. It's very costly. Usually it has to be personalized because every one of us can be immunogenic if you take your cells and put in other people. So there's really a big, big issue here. There's also another important issue, which is delivery. How can you deliver this kind of engineered synthetic cells, or even mRNA and DNA specifically in your target tissue? And this is particularly important if the tissue that you want to target is kind of encapsulated. So it's much easier to deliver things in the blood. It's much more difficult to deliver things in the brain, for instance. Also, if you think about gene therapy, you take your curative, potentially curative transgene, you want to put in a viral vector and deliver to the brain, but if your transgene is too big, you cannot encapsulate it. So there's also a problem of transgene length and also transgene regulation, how tightly has been the transgene be delivered in the synthetic cell. And there's always a problem of toxicity. We're talking about live cells, how toxic can be these live cells when you put them into patients. And finally, personalization. As every kind of drugs, even for gene and cell therapies, each of us can answer differently to the same kind of drug. So these are the general problems we are thinking about in the gene and cell therapy space. And now in the next 3 slides will be 3 different case studies of how actually AI can help to solve these problems. So one is the manufacturing problem. I want to point out this very interesting company. It's a new company in Silicon Valley. It's called Multiply Labs. Basically, they are making AI robots that can help and speed up the manufacturing process. So it's like a very preliminary, but from what we are seeing, they're kind of building some bioreactors where you can expand your cell therapy, and then a robot, like think about the Tesla robot, but like an AI-based robot for cell manufacturing. This should speed up and, let's say, decrease tremendously the amount of cost in cell therapy production. These other case studies about delivery. So how can you make tissue that you want? There's this company here, Therapeutics, what building is like libraries, agnostic libraries of viral vector, each different from the other, from the capsid protein, and try through high-throughput screening, match with machine learning, trying to identify which is the specific capsid protein, engineered capsid protein, agnostically wise, that has to be used to deliver the cell to the specific tissue. And finally, a last case study, how can synthetically generate de novo regulatory sequence that make your transgene be expressed just in your specific target tissue? This is a new study from Genentech in which they were able to synthetically identify and design from scratch regulatory sequence that can make your transgene expressed specifically in the liver, from red blood cell and brain. So again, if you want to think about gene and cell therapy application, potentially different from what we have seen before, please reach me out. Thank you.
Thank you, thank you. Okay, so next up, we have the co-founder and head of research at NewLimit, Jacob Kimmel.
My name sounds so much better when you say it. I'm going to record that so I can use it later.
Hi.
Hopefully this opens. Great. Thanks all for coming. Thanks so much, Jeremy, for the invitation. I'm probably going to give one of the laziest talks you'll see today, which is because I did not make a custom one for this event, so sorry about that. What I will run through is a bit about the problem we're working on, and I'll try and generalize it a bit to the more general problem of drug discovery and leave off with a few highlights of things you could work on today. So at NewLimit, we're focused on trying to extend human health span. I like to joke this is really the motto. It should be the motto of every therapeutics company. We just like to say it out loud. So what we're working on very specifically, and I'll ask you to trust me on a bunch of these points so we don't take up too much time, is trying to find combinations of transcription factors, these are specific genes, we can activate that when they do so are able to change the age of a cell. So we actually know this isn't totally science fiction. We can reprogram cells just by turning on combinations of transcription factors, and we can reset both their age and their type simultaneously. This was the discovery of induced pluripotent reprogramming factors by Shinya Yamanaka, won the Nobel Prize, really awesome science. We also know you can turn cells across different types by just turning on transcription factors while preserving their age. So I can take an old skin cell from an old person, reprogram it to a neuron, and it looks like an old neuron. So we've got this 2D coordinate system here, and we've got existence proofs in both the diagonal dimension and the y dimension. NewLimit is trying to figure out whether or not you can navigate just along this x dimension here. Can we reprogram cell age without changing a cell's type? This would be necessary if we actually want to make therapeutics. Unfortunately, while we have this cool science experiment we can run where I can turn a cell back into an old cell back into a young stem cell, I can't do that in a body. That would cause teratomas, would be very bad. However, if we could reprogram just the cell's age, potentially you could take an old dysfunctional liver and make it more functional, an old dysfunctional immune system and make it more functional again. So the challenge we're trying to go after then is, can we find combinations of these transcription factor genes that are able to achieve that goal, just reprogramming cell age? As Atray nicely teed up, the challenge that a lot of those with CS backgrounds will recognize immediately is that when you have combinations, immediately you run into a place where there are far too many hypotheses to test in the real world. So 10 to the 16 may not sound like a crazy number to those of you who deal mostly with CS problems day to day, but this is like far more experiments than anyone has done in any regime in biology, period. So you're not gonna be able to just experiment your way through this problem. So we need to be able to design experiments that, yes, cover as much as we can, but then more importantly, figure out which of those experiments is worth doing. So we've thus far only looked at cartoons, and we'll largely just look at more, but I want to give you a little bit more texture on the type of data we generate to try and navigate through this space and figure out if this problem will be tractable. So what we do at NewLimit is actually not so distinct from some of the challenges that Atray mentioned earlier some folks are using with similar single-cell screening regimes. So we start with young and old cells in our case, but you could imagine in the more general setting, this is diseased and healthy. We deliver combinations of perturbations. In our case, we're focusing on these transcription factor genes. Happy to talk about why later. And then we can turn these on briefly, mimic what would happen with a medicine, and we can collect a profile of all the genes expressed in those cells. So what you get out of that is this nice matrix of counts of how many times you saw each gene in each cell, and then a matrix of labels, which things you perturbed them with. And the question you want to ask is, when I start with these old crummy cells, do any of the combinations of perturbations make them look like the younger, healthier versions? That's just the start of maybe making a medicine, but helps you point in the right direction. Now you can do this for thousands, tens of thousands of combinations. You can't do it for 10 to the 16, or if someone knows how, please let me know. So as Atray mentioned, the actual amount of this data this is a graph for our specific regime, but this curve is pretty similar regardless of the specific target space you're looking at. The amount of this data that existed just a few years ago was pretty paltry. You couldn't really build useful models. But as just a small organization, our company is only 30 people, we're able to 300x the amount of public data over the course of about 18 months. So you can start to understand that this is an area where the data is inflecting at a rate where it starts to make sense to apply models. So I'll give you just the briefest notion of how we're trying to frame this modeling problem, and then leave off with some of the sub-problems that honestly some smart people with a lot of Red Bull could tackle in a weekend. So two of the preliminaries for us are, if we want to be able to train a model to predict which of these hypotheses should we go after, which combinations of transcription factors might make an old cell look young, hopefully eventually act young, we need some way to first represent what does the starting cell profile look like in a computational and quantitative way. So this is a representation learning problem of the cell state. And we also need to be able to represent those targets we're going to perturb, the transcription factors. And the nice thing is that we've got reams and reams of data on both of these things. NIH has sunk billions of dollars into learning a lot about transcription factors. So there's lots of data out there on the open web one can use to try and learn better representations. We then get to the fun part, which is you want to take a model that can take in both of those representations, I'll call them tokens here in our universal parlance, and try and predict what would that do to the expression profile, what genes would go up, which genes would go down, does that cell look younger or older, healthier or more diseased, and maybe if I even have some annotations, do I think it'll function better? And then ideally, you could wrap that whole thing into an active learning campaign or a Bayesian optimization-like campaign for those of you who are more familiar with optimizing hyperparameters in this fashion. You've got a limited number of experiments, and with a model that predicts which of them are worthwhile, can you use that budget effectively? So this is a hypothetical and idealized version of what these models might look like. It's all cartoons, as you'll notice. So I want to leave off with just some of the problems that, again, smart people one weekend could probably make a dent in. So the first is that we don't honestly have great quantitative representations of TFs today to predict combinatorial effects. This is something where all the data is out there, but until fairly recently, you didn't have enough of these perturbation data points to really make this problem worthwhile. So there's a couple data sets here I'm just flashing, so I think you all have a link to this talk as well you can get, that you could go download and try to use some of the public data on the internet to learn better representations and then predict what effects they had in a couple of these screens. Another is that the way we represent single cell profiles for use with modern, large, self-attention-based models is honestly not that great. No offense to anyone who's built these before, but we do really silly things. I'm pretty sure if someone spent some time trying to be clever in how they tokenize a profile so you can feed it to one of these models, you can substantially improve on some of the benchmarks that have come about. And then finally, one of the nice things in biology is that we actually do collect data in a variety of different batches, and every experiment has both the real causal covariation mixed into it, as well as some just purely correlative covariation. So if any of you were at NeurIPS in 2019, you probably remember all the hubbub about this notion of invariant representation learning. If I collect data in a bunch of environments, I can start to understand causal versus non-causal effects, build more general models. And a lot of those talks and conversations in NLP and computer vision dropped off with well, we don't really have any alternate multiverse world cameras going around. We don't really have other variants of the English language that evolved in another timeline. So there's no such thing as a different domain. In biology, this is kind of always the setting that you're in. You always have different environments that mix this causal and non-causal information. So trying to use some of that data to learn less variant representations that eliminate those effects from what we call batch effects in biology, but retain the real underlying graph of what's happening and what gives rise to the cell state you care about is another one that you can make progress on in just a couple days. With that, I'll leave off. We hope to extend all your health spans.
The generation of 300x more data, do you ever get pushback, whether from customers or not, on the accuracy of that data, and if you've checked it with experiments?
Yeah, absolutely. So this is basically a validation rate. So what we do in these screening settings, and I'm getting a little into the weeds here, NewLimit's developing mRNA medicines like those that Krishna is optimizing. And so we're trying to mimic that in one of these genetic screening regimes, which mRNA medicines aren't really amenable to testing tens of thousands of things today. So we're trying to build a Potemkin version of an RNA system that we can then use to predict what will happen. So we do get people who ask, how well do these things validate? If you discover something with your clever genetic tricks, do you actually get the same effect when you deliver an mRNA? And the short version of that answer is, works pretty well, and so it's not something we worry too much about. But we did have to spend a bunch of time engineering our system so that it actually predicted what would happen with a real drug-like product.
Is there a consensus for how cell age is defined, and how would you get at measuring cell age in a high throughput manner? Is it methylation patterns or something else?
Yeah, awesome question. First answer, no. People say all sorts of things when they mean cell age. You read the literature, there are a bunch of really bad definitions, like how many times you passage the cell in a dish, or how many times you irradiated it. I think none of these things are aging. Aging is what happens when a cell sits in your body for close to 10 decades and weird stuff happens in its environment. So for us, the way that we try to learn a representation or a quantitative notion of cell age is pretty darn simple. We went out into the world, we collected a bunch of cells in each of the cell types we work in, hepatocytes in your liver, T cells in your immune system, from really old people, really young people, and everything in between. And then you collect expression profiles from those. You could ostensibly use another modality. We actually experimented with a number and found that for the money and the bang for your buck, RNA gives you most of the value and is pretty darn cheap. And you can then train a model to predict age. And you can do this in all sorts of very challenging regimes of, I'm going to hold people out, donors that the model has never seen before, make sure I can predict age. And what I would say is you'd be shocked with how easy that problem is to build a model for. It's actually quite trivial if I take a draw of your blood to tell you roughly when your birthday is, likewise from your liver.
Are you looking at DNA damage at all? Because reprogramming changes the gene expression profile, but there's nothing about the DNA mutations that can't.
Yeah, amazing question. Short answer, no. We at NewLimit are not focused on trying to fix DNA mutations. Those are another component of the problem of aging and something that our medicines will effectively, in the current regime, never treat. So what we're hoping to target are areas where just changing the expression profile is sufficient to impact a patient's disease, give you a better overall wellness. But we're not, for instance, going after right now trying to reduce cancer incidence by reducing mutation rates in a given cell type or something like this.
I was wondering if, given that you have a large search space, like the combination of prescription factors, whether you will be amenable to using prior information, like, for example, there's knowledge about chronic inflammation driving aging, there's connections with metabolism and metabolic stress affecting aging, and obviously, that also depends on your definition of aging as well. But I was kind of curious if you guys are using those strategies to narrow down the search space.
Yeah, it's a great question. So short answer, no, we don't have like some quantitative encoding of a lot of these priors, like what is the chronic inflammation environment like. What we do is we try and validate everything we do, even in some screening scale, in vivo in animals. We don't feel that we're smart enough to cleverly encode all of the different inputs a cell is receiving in an aged organism. So let's just try and test some things in an aged organism, treating that as like the best model of what aging is actually like, and then determine whether or not things that we're able to discover in vitro in a dish or in a non-aged organism actually replicate. So that's the way we're going about it.
Sorry.
Yeah, with that, I just want to make sure to get on the other speakers.
So much thanks for all of the concrete hackathon projects. Let's hear it for Jacob. Okay, no less a great Jacob Rinaldi, the co-founder of Noetik, is here to grace us with a number of ideas. Let's hear it for Jacob.
Cool. Okay, so why is drug discovery hard? Why do most clinical trials fail? Really, our thesis is that the answer to that is simple. We don't understand human biology well enough. So in a cubic millimeter of your cortical tissue, there's 150 million synapses, and in our domain, immuno-oncology, we're dealing with this milieu of cell types engaging with an evolving tumor. And typically how we try to grapple with that complexity is by building simple models. Let's find one gene that's going to predict the response to this drug. But that's really how we wish biology worked. It's not how biology works. And so from our perspective, the promise of machine learning is grappling with that complexity, grappling with the intrinsic complexity of human biology, and pushing that back into the drug discovery process. And so to this end, we are building a full stack drug discovery company centered on machine learning from first principles, but the initial focus is the hard problems at the start of the process. Which drug should we make for which patients? And so first we have essentially a laboratory in South San Francisco generating large-scale training sets for ML. We feed that into custom vision transformers that make therapeutic predictions. And then we have a large-scale in vivo screening platform that we in-license from Mount Sinai using CRISPR. And so, what do I mean by a data factory for self-supervised learning? I think the lesson from our previous company, Recursion, was that typically when people say this, they mean we're going to do 20% more than what's happening in academia. We're going to use data collected during standard clinical care. And our perspective is that's really insufficient to train machine learning models. So we have built a laboratory that takes human tissue sections, uses a machine learning
algorithm to sample from them, builds arrays of those tissues, and then for every patient generates a multimodal tissue stack in batches that are randomized alongside controls, proteomics, transcriptomics, whole exome sequencing, H&E. And then we take that data factory and we feed it into custom vision transformers that train on the entire multimodal data stack. So they are essentially learning to generate portions of the data set from everything else in the data set, essentially becoming foundation models of cell and tissue biology that train on very large-scale raw human data. And so crucially also these models are designed to be both interpretable and queryable, where by queryable what I mean is that I can change any aspect of a specific patient's biology. I can change one oncogenic alteration for another. And then I can ask the model, okay, in that sort of hypothesized alternative universe, what else about that patient's biology is predicted to change? So as you can imagine, this becomes a system for large-scale in silico experimentation that we can then push into animal models downstream. And so here's just a snapshot of how that works using known targets. So on the far left is a large-scale screen where we asked the model to find things that would induce T-cell infiltration. One of the top hits was AXL, which is a gene known to induce this phenotype. Crucially, the model understood or thought that that was dependent on another specific part of the patient's biology, whether or not they had a KRAS alteration. This was not in the literature. Then we can replicate that effect in our in vivo system. If we knock out AXL, we get T-cell infiltration. And then this is a public clinical trial that wasn't designed with this KRAS hypothesis in mind, but you can see all the benefit goes to the KRAS patients. So yeah, I guess my take home for the hackathon is like, let's fucking go. Like I think we're seeing stuff every month that I didn't think I would see in my career. It's a really amazing opportunity to impact human health, but like, it's not gonna happen if people don't do it. And so, yeah, take advantage of this moment. Thank you guys for your time.
We're seeing things every month that I didn't think I would see in my career. That sounds exactly right. So I am going to give a brief presentation about Aperture. Okay, little self-introduction. So my name is Jeremy Nixon, the founder and CEO of Omniscience. And Omniscience generates deep, grounded writing. So everything from cited research papers to Amazon bestseller books. So if you want to peruse some of the texts that our customers have created, they're to my left, and legal defenses. This legal defense in particular was the center of really intense political controversy, and so 300,000 people have read this AI-generated, cited legal work around SB 1047. But in the context of the biohackathon, We have a specific gift for you all, so everyone will have as many PaperGen tabs as you'd like. We'll give it out, and we'll have a $200 prize for the best use of Omniscience today, because we produce cited research papers grounded in things like a GitHub repository, where you give us a GitHub repo, we discover the subset of the functions in that repo that are novel and of interest, and we write a research paper that's guided by you, that's grounded in the work that you've done around an important subject. And so I'll give you a quick glance at what that can look like for you today. We've built an AI that can write the first draft of your research paper. So let me open up, well, it's kind of hard to see. Well, I guess. I'll have to come up with the size. Hopefully it can be functional. We've built an AI that can write. We've built an AI that can write the first draft of your research paper. This is an example of a paper created yesterday about our new BCI dataset release. It's flush with dozens of accurate citations and relevant mathematical analysis. The set citation set can be seen here, and you can see the quality of the analysis here. I put a description of the work that we did into the Omniscience paper generator by going to omniscience.tech/research, adding the description to describe paper, and adding the authors and our institutions into the author space, and click write. This paper was emailed to me in less than 10 minutes. The email contained the PDF of the research paper, as well as the LaTeX so that our team could start editing. We used Overleaf and added the style file Omniscience sent to us. And just like that, we were off to a running start with our research project. To use it, go to omniscience.tech/research. We give you a generous 2-week free trial. Thank you. Okay, that's Omniscience. So the simple frame is basically, we will sort of recursively call prompts on the subset of text that you've given us, or code that you've given us, or data that's being analyzed, and can do visualization in loop. So in a lot of cases, you have a writing industry where you're trying to, put together a visualization set, a table set, an algorithm box, basically all of these artifacts in your research paper. And the content from the algorithm box is typically inside of your project's GitHub repository. The content for the graphs that are in your research paper typically come out of your data set, and the data set is being analyzed. Certainly there's some transformations that happen which can be executed with code generation, and then you can have a code generator create a visualization that's the basis of some conclusion or item of interest in the paper. And so we are going to make it easy for you to basically give us the raw material of your research process and get to a first draft or a publishable example of what you've done. The second thing is lit review. So if there's anything in literature review that you'd like, which would be customized, where there's basically a very specific question that you have. For me this morning, I was really curious about how much, in the particular subfield of machine learning research, automated machine learning research I'm doing, how much code generation has been a part of automating research processes. And so I can get a lit review with every paper that's on that subject just by requesting it from the system. And so we're going to send out a basic link. Do try out PaperGen on projects of interest. Sometimes if you discover this, as some researchers have basically said, oh, well, I have all this unpublished work. And the reason it's not published is the process of writing their work up takes a really long time. The process of finding all the relevant citations that show the history of the work that is behind theirs is really laborious and time-consuming. And you can use search in combination with generative models to discover the appropriate citations and include them in a paper. So we'll send it out. Do enjoy the Omniscience. Okay, and with that, Sophia Lugo, the co-founder and CEO of Radar Therapeutics, will grace us with a presentation.
So while he finds that, I also did not prepare something particular to this, but there's so much that AI can do that I'll talk about a use case that would be really cool if somebody else figured it out so that we had to do less experiments in the lab to validate. I think I just...
I can send it to you again.
Let's go.
So I'll start with talking about what we do at Radar Therapeutics. At Radar, we make programmable, precision-expressed mRNA therapeutics. So as Alessandro mentioned before already, it is really important for, if you're reprogramming cells for any genetic medicine, whether it's mRNA-based or DNA-based, that you're only reprogramming the right cells in most applications. So that's what we do at Radar. We make mRNAs that are conditionally activated based on the presence of a transcriptomic marker. So this is our team, so I have to credit these guys for inventing the mechanism that is foundational to the company, and it doesn't represent all the people that are hard at work at the company to develop this. Yeah, so at Radar, we believe that exquisite specificity is the next frontier in medicine. It has been very difficult. A lot of people are working in the delivery vector space. They're trying to do, as I mentioned before, capsid engineering. They're trying to, on the LNP side, they're trying to reformulate LNPs or decorate LNPs or change the charge on the particle to try to get them to not only deliver to the right tissues, but then furthermore to deliver to the right cells within those tissues. And that problem has been extremely hard to do. It's been extremely hard to do, particularly in a rational way. And so at Radar, the big innovation we have is instead of thinking about limiting entry to the cell, which often is based on a cell surface marker that you try to interact with and then specifically enter a cell, why don't we actually make the expression of a genetic medicine conditional on the direct code that tells you more about what cell type you're in? And for us, that direct code has been the RNA profile of the cell. So the DNA broadly same in all cells in your body, but what really tells you about what protein profile the cell is going to have is the RNA profile of the cell. And there has been an explosion in single cell transcriptomics, and so if we could, in theory, just read the RNA profile of a cell, you should know what cell type you're in. We take this fact and have made our mRNA's expression conditional on finding a transcriptomic marker or an RNA signature that defines a cell type. In that way, you can have systemic delivery, so you just distribute everywhere, and then you really make sure that you only turn on when the code tells you you are in the right cell type. And if you could add, this is essentially adding an if function to an mRNA. If you find this marker, only then express. And if we could do that, it opens up a slew of possibilities. You're now able to look at the code, so the RNA marker for an undruggable protein, for example. And based on this marker, so it doesn't matter if it's secreted, it doesn't matter if there isn't a good functional interface, but you find this marker for an undruggable protein and you make it druggable, or you drug it directly. You could find markers for fibrosis or aging, and then using those markers express something that specifically regenerates those cells or inhibits a pathway. You could find cells under attack and specifically protect them. You could find markers for cancer, or even markers that are highly correlated with an oncogene, and then specifically silence or eliminate those cells. Lots of possibilities. So how do we do this? So essentially what we've created is a logic-gated mRNA. And so you imagine this part is the part that encodes for the therapeutic protein. Upstream of that, we add a stop codon, and then a sensor region. And I'll talk more about the sensor region. But of course, we said at the beginning, what we do is we use bioinformatics to find a transcript that is highly differentially expressed in one cell type versus another. And really, we're looking at the universe of cells that you deliver to. And then what we're trying to do, the name of the game is this stop codon will inhibit the therapeutic protein from being expressed unless it gets unlocked. And so to unlock this, what really has to happen is it has to hybridize with the, so we encode a reverse complementary sequence here that will hybridize with the transcript of interest, and only then does this get unlocked. And that happens because an enzyme called ADAR, where the name radar came from, will come in and inactivate the stop codon. So I'm trying to just run through that quickly to get to where I think there's a lot of opportunity for AI to work. And so, you know, the ADAR space has been exploding. A lot of these companies have been producing actually a lot of good data in the clinic. So on the public side, I'd invest in these stocks now. They're all been on the rise. But the way that the ADAR companies work is essentially they use this property, ADAR, this enzyme, and I think I'm skipping back and forth through a lot of things, but what this enzyme does is it very specifically makes it takes in a double-stranded RNA, it makes it change from an A to an I. So it'll come in, find double-stranded piece of RNA that's spuriously formed in cells, and it'll change an A to an I. So these companies are essentially ASOs that are changing specific, where there's a specific G to A mutation that needs to be changed back. And an I looks like a G, so it needs to be changed back from an A to a G. So for these ASOs, or for a company like ours, like Radar, ours is different in that we're just looking for a target gene, but we're not trying to fix this endogenous transcript. We're just trying to make this hybridization region bring in ADAR and inactivate the stop codon so that this can get expressed. So without further dwelling on what the company does, a lot needs to be done to get our construct to work very reliably. Some of that is predicting how the specific sequence context here on the payload side ends up giving us really robust expression, or ends up with a less immunogenic synthetic mRNA. And a lot of that work, a lot of advancements in structure and function prediction on the mRNA side have been done directly by Amit at Deep Genomics when he worked on big RNA. So I won't talk about that. Another challenge we experience at Radar, or any company that's working in an ASO, or any company that's working on a small molecule that's drugging RNA, is that you really need to know a lot about what's going on with these guys, endogenous transcripts in the cell. So the endogenous transcripts in the cell, in theory, it's a direct code. The code is telling you where you should hybridize. And you should be able to, in theory, hybridize with any transcript that is reverse complementary to the sequence that we've encoded here. But in practice, these transcripts act really differently in different cell types. They might have different secondary structures. They might be found in different subcompartments of the cell that affect how easily we can target them. They might be folded in ways that make them inaccessible for us to hybridize. They might be interacting with several proteins, RNA binding proteins, that don't allow them to be accessible for us. And while there are many labs doing experiments trying to understand which RNA binding proteins affect different transcriptomic sites, so different sequence contexts and different site motifs, it's been very hard to have a universal profile of per transcript, per cell type, what is the universe of RNA binding proteins that are interacting with that transcript. And that's super important, because RNA binding proteins, if we know that there are proteins interacting with this RNA site, that can tell us a lot about the availability of that structure. Either an RNA binding protein interacting with it can tell you this is actually a really good site to hybridize with because the structure is that you can access it, or it can tell us this site is actually blocked by that protein because the affinity is really high. This is all to say that, in theory, this can look like a very simple and elegant switch. Very simple and elegant. You just inactivate a stop. But this hybridization event is actually really complex and difficult to achieve without understanding what's going on in the cell. And unfortunately for RNA, we do know that as you move between different tissues, the RNA starts to behave differently and compartmentalize differently. It might hang out near different organelles or in different organelles, and that will affect how you decide to deliver it or where you decide to deliver it. So we could take a whole brainstorming session just adding on the different layers of complexity that you have to think about for every gene you're trying to target. How is it folding? What are the proteins that are interacting with it? When you perturb the cell, does that change? How do different genetic variants change that? It's very hard for humans to do that. And in the lab, for us to validate that is very difficult. When we talk about the cost of experiments, experimental validation is typically what takes so long. If there was an in silico way to say, hey, for this given gene in a given cell type, these are actually the regions that are accessible for us to target, there would be huge advancements for ASOs, for radar, for small molecules targeting RNA, for everyone who's trying to interact with these guys that tend to fold very differently and interact very differently with their cellular environments per every cell type. So yeah, I hope that made sense, but the idea is that the level of complexity in biology is extremely high. The huge opportunity in deep learning is obviously that a computer would be able to intake all of these different components that we have to think about, understand the literature that's out there, and come up with a global profile of protein-RNA interactions, and a more thorough understanding of what happens when you change between different tissue types, and even when you move from in vitro data to in vivo data, which has been a huge challenge in any data sets generated about RNA-protein interactions or RNA structure and function prediction. Thank you.
Imagine a version of this company that's using like ML plus promoter and enhancer sequences to try and get specifics up. Is that just like a shitty idea? Is that too leaky? Like, do you need the lock, or like, how do you think about that?
Yeah, so for promoters, so one, a version of this company that uses promoters, you probably want to be in the nucleus. And so we imagine that there are actually a lot of advantages to doing direct mRNA delivery. So that's one advantage. Another for promoters has been like, if you're using promoters and you want validated delivery, you're probably using AAV. And AAV has limited loading capacity, and some of those promoter regions in the brain are pretty long. And so you start to get challenges with promoters where when people have tried to shorten them, they're not very good. And so their promoter engineering is, and many technologies are trying to accomplish the same thing that we are. We believe there are advantages in working directly in mRNA. There are advantages to the size of our construct. It's very small. It's like 100 to 500 base pairs we add to the gene, essentially. And so, like, yeah, all these technologies are worth working on, but for ours, yeah, there are versions of those technologies out there, but we see advantages in selective activation of mRNA and our size.
The next question's also mine, but do shout out for after that. Thank you, Sophia. That's right, give her a round of applause. Wonderful. Now we have the co-founder of Deep Origin, Natalie Ma.
I will not have any, there are no slides. Also, how many of you like want a second to like stand up and just sort of like shake it out? Like, seriously.
Get up!
Come on! Just sort of like get your blood flowing again, because your brain is just like fuzzy from all the heat. I am the last person, and I'm going to raise Sophia and Jimmy even one more, and I did not even bring slides. I don't bring slides for these kinds of things. It's a little more fun to have an interactive session. And to kick it off, I want to just ask the question: how many of you are what you would describe as like very AI first versus bio first? So like AI first, raise your hand. Okay, now bio first, raise your hands. Okay, good. Actually, this is a more even spread than I thought we'd have. This is excellent. And then Amit gave a great introduction earlier for like drug development. How many of you have had experience taking a drug through from discovery to development to approval? Like discovery, discovery, development, approval. I was gonna say just you probably. So really quickly, I'm going to talk a little about that process and like what that entails, because I think that's useful for folks to understand. Quickly, my background, PhD in synthetic biology, working on the Jurassic Park problem. How do we engineer organisms, put them in open environments, and not have them go haywire? And then worked in management consulting, went back to my PhD school, helped them launch startups, got several startups off the ground, and then jumped into those as a serial co-founder, operator, mostly on the business side. So I'm going to have a couple of principles primarily for the day after today, when you're thinking about, do I want to be a for-profit entity? Do I want to be a non-profit entity? What does that look like? And then a little bit on the principles of, how are we developing? Why are we developing these things? So as Amit had earlier, this pathway flowing through from like discovery to development. Everything that we are doing in drug discovery through development is an approximation of how this is going to work in the real world, right? I think a lot of the folks who are in bio probably realize this, but like we start at the purely abstract level of in vitro binding of some kind, protein totally separated from what it is doing, and then move into cells, and then move into mice, and then move into monkeys, and then move into people in increasing populations that we think will estimate the real world. And each of those has tremendous inaccuracies associated with it. So for the folks who are working on data sets today, it would behoove you to never forget that and ask the question of how much does the data set that I'm looking at reflect the real world. Cool? Awesome. I don't know if I'm going to fulfill your role of actually having an idea for AI application, but this is, I think, another good point is like each of these details, as you go into any given space, right, you have the big high-level idea, chances are you don't have a solvable problem. You get deeper and deeper into specific problems, right? So as was brought up earlier, you have the ability to make a drug. One of those is affinity. One of those is tissue permeability. One of those is how quickly is it urinated out of the body or expended in the feces, right? Each of those is a different problem you have to solve, right? And each of those is probably not going to be a unified model, although I guess they could be proven wrong, but its own specialized model that you have to think about. So that's more of a plug to say, for the folks who have laid out problems here in their startups, those are phenomenal problems to work on because an expert has already defined a space for you to work in. And then the things sort of on the day after. So like I said, I work primarily in business development now. I was a scientist. I will say that there has not yet been a successful model for software in science. Just gotta say that, right? I know that people get very excited about it, but if you look at the winners in the space, it's when you apply something to a drug, or you have a more general problem than making a drug, right? So I think things where you develop papers for legal as well as for science, you've got an opportunity. But ultimately, if you're just doing price times volume, price has to be super high, which makes it a lot of, for a variety of reasons, you can come reach out to me afterward and I can explain, super hard to sell, or your volume has to be higher than the number of actual scientists working in drug development. So think about that, especially if you're thinking, I want to create a for-profit entity after this, right? Like, where is there actually money flowing? Who will give you money? And then where can you make a successful exit? And then sort of lastly, in the time that's sort of running out, I think one thing that doesn't get talked about enough in a rational sense, usually it's an extremist or like world is falling apart sense, is what is the ethical responsibility we have? What is the moral hazard of what we are building? And this tends to get very quickly into AI is going to turn us all into paper clips, which are like too far off the side. But like if you just think one step removed, if this gets released, what is the effect of this? Great example is somebody created a model developing antidotes to poisons. It turns out that model was great at generating poisons, right? Like that is a way you can use the model in a nefarious purpose. Like as you think about what you're building and working on these problems, how could these things be misused? Not 8 steps out, just 1 step out, right? Like think about that and then think about, is there anything you can do to mitigate that? Because I'll ask the question, who thinks social media was deployed responsibly? All right, well we have one person, but generally, like we feel like that was technology that was put out there. Would we want biology to be that way? Because if the social media generates animosity and circles, what happens when biology is misdeployed? Somebody dies, right? So that's a very different world. So I feel like I'm ending on a very, very dark note. Apologies, Jeremy. I don't know if I'll get invited back. But those are just a few things for you to consider, as you're building, not just the technical side, but what value are you creating for society and what does that balance look like in the world when you're done with your project? So thank you so much again for the invitation. I am so excited to see what all of you are going to build. And hopefully, you change the world with what you build, right? Because there's so much going on that we never imagined was possible with data today.
So yeah, I guess that inspires me to speak for a moment about the inspiration behind this event, which is actually about impact. So I guess I spent a lot of time in communities that worry about things like paper clips. So I guess if you ever wonder about the origin story of OpenAI. In part, it's that Shane Legg, who had been in the rationality community, and Demis Hassabis founded DeepMind, and Elon was afraid Demis was going to destroy the world with artificial intelligence, and so decided to create OpenAI. And they had a schism, and years later, where Anthropic came out of similar extinction risk kinds of question. And so, yeah, I guess more recently, literal safe superintelligence is the name of Ilya's new great AI company. So it's sort of been a central theme. Now, thankfully, the risks have stayed hypothetical, at least thus far. No one has died, or at least for the most part, things have gone relatively well, which is fantastic. I would say the social media revolution in comparison to the AI revolution has had a lot more sort of consequences, and AI seems a lot cleaner. But I do think it's really important to appreciate that P-doom, which is the conception of the probability that everyone dies, can be reframed as P-life, which is the probability that you and I are still alive, in N years, and which incorporates the possibility of apocalypse, but also does offer the upside of health span and lifespan. And so my hope is that the technology being present in biology actually opens up a lot of real benefits that are quite deep and core to who we are. Whether it be in brain-computer interfaces or in, as I said, extended health span, or in other sort of ambitious ideas around how to integrate with artificial intelligence to do prosthetics, to advance bioengineering, to create protein folding models that are sort of downstream tools for the rest of biological science. Obviously, umpteen opportunities abound, and so I see a lot of hope in an event like this, and that's part of why it exists. So we're gonna have one more presentation. It's from someone who we met who was deep at Google search. So they were the one who launched the retrieval augmented generation system at Google, and they just left to create new data conditional tooling and have built a product specifically for you all today. So let's hear it for Karthik.
Yeah, thank you so much. Hello all, good morning, wonderful to see you. I'm Krishna, I'm co-founder and CEO of Data Bootstrap. Until 3 weeks ago, I was a researcher at Google DeepMind, and very exciting things happening outside for me to not miss it. So I quit and created this company. A couple of days ago, I was having a chat with Jeremy, and he mentioned, hey, you're doing something with data, search, chat, ML, and all these things. Why don't you just index all the bioarchive articles and papers for this hackathon? So my co-founder and I, foolishly enough, took it upon as let's do a hackathon for the hackathon. And for 36 hours, been coding straight, trying to load all these things. And you would, some of you would know how difficult to do this large-scale things when you do not have a big cloud at your disposal. So whereas I wouldn't have blinked while I was at Google, 350,000 still took a lot of effort to get things, but yes, 350,000 research papers, all from bioarchive, and to be honest, most of it is still in loading, so fingers crossed. But we created this API for you, but before that, a general thing, there are only two kinds of people in the world, those who have a data problem, and then the rest, those who do not know they have a data problem. So we thought, okay, we'll provide this data for you. It's a very simple API compared to all the other biology stuff. This should be easy peasy for you. You pip install the thing, get yourself a token, register in the site Data Bootstrap. We would appreciate it. And then you send your query for a chat, or you send your query for a search. And you get all the relevant results from the bioarchive papers with a link. And you can then further get more text to add to your other applications. So eagerly looking forward to how you would be using it. I'll be here. Feel free to ask me any questions. And I'll also post these links in our in the Slack group as well. And yeah, please be gentle on us. I haven't even showed this to my friends yet. You're all the first. I didn't want to show to them something done very quickly, and they're going to look at me and go, you quit DeepMind for that? No. So, but you're all hackers, so you'll be more patient with us. So this is the first official launch. I mean, kind of a forced launch. Thanks to Jeremy. Yay. There you go. Thank you so much. Oh, you have a question? Oh.
Can I just ask a clarification question so we can better use this? Are you searching for exact words and terms, or is it smart enough to understand neighborhoods?
No, great question. So it does the semantic search and keyword search a little bit combined, but for the most part, the semantic one for now, and so it's a box of chocolates. You have to try what you get out. Honestly, I'm not like too sure about. I'm like a little bit nervous, but we'll find out. It does like both with the embeddings. Yeah.
Is it reading charts?
Oh no, not yet. Oh, that killed us. So I had to like comment out the multimodal and image part and do only text only for now. Yeah, cool. Hey, thank you so much. Yeah.
Okay, that's it for our speakers. Let's have a big round for all of the speakers today. So this is the point at which we turn the mic to the hackers to propose hackathon projects. So if you have a project idea and you'd like people to hack on it, do feel free to jump up and present it. And I guess Calvin, I think, also wants to present Idea Flow. You guys have a tool for the hackathon to use. But if you're going to give a talk, do line up here so that everyone's sort of prepped to go. So Justin, Elliot, Eric, Manu, Karen. And if you are interested in giving a lightning talk, but if not yet signed up, do join them to give a talk. Yeah, if you like. Do you have a presentation, or do you want to just speak?
A few slides I just put together.
Yeah, you just.
Hello. Hi everyone. My name is Calvin Xu. I'm an undergrad at Stanford, and I work with IdeaFlow, where we work on helping people massively collaborate on difficult problems. And the main thing we're building is a next generation knowledge base and ontology editor. But as a side project, we have something pretty cool working on that is kind of complementary to what Krishna just showed that I think will be interesting. So Krishna's tool was on bioarchive. We actually built a knowledge base, knowledge graph that ingested the 2023 June baseline of a ton of PubMed papers. So currently in our Log4j database, we have over 40,000 entities, as well as the relationships between them. The relationships are derived with few-shot GPT-4 prompting, and you can write queries to see, such as in this example, the shortest path between all of the IGF2 genes and also ovarian cancer. And the idea started when everyone's very excited about the performance of large language models on knowledge graph discovery. So we actually started building synthetic data sets and also benchmarking how well large language models perform on these tasks. And what we find is that, of course, it massively surpassed all previous unsupervised methods. And I guess the other thing I want to briefly show is this brief demonstration of what's possible with the knowledge graph. So this was something I was working on with a causal inference lab at Stanford. We were trying to do a genomic perturbation prediction task using a perturb-seq data set. So the author is actually with us here today. We're very honored. And so what we're trying to do is that there was an existing causal pipeline that takes in a lot of data and generates a DAG of all the causal relationships. And we're trying to hopefully augment that with the domain data from PubMed. So this was the result of combing over basically all the genes that were in the genome that we were interested in. And this was done by, we were submitting a query to match the shortest path between these genes. And we printed out, all of the relationships between the intermediate entities on that path. So when we collected everything, which was over 60,000 paths, we then did a second pass with GPT-4 to let it tell us if it thinks that that is actually a regulatory pathway. And so we ended up with a smaller subgraph where the nodes are kind of sized according to their algebra. And we plugged this back into the causal pipeline, which improved the performance somewhat, but we really think we ought to move on to a deep learning approach by now. But yeah, so that's just, we think, an example of what, knowledge distillation and what's possible with our project. And we're excited to find more domain experts who are willing to try out this project, because we definitely don't know enough bio to, I guess, capture the full potential of all the PubMed data. So thank you very much, and we're excited to work with people today.
Very cool. Okay, lightning talks are going to run for 1 minute per. So first up, Justin.
All right, I have to plug this in. I have one slide since it's 1 minute. All right. While this loads, I guess a brief background. I'm a researcher. One of the things I'm working on is discrete diffusion and specifically adding guidance. One great application for diffusion models, and in particular discrete diffusion models, is protein sequence generation. And the reason why adding guidance is very important is because, as many people have mentioned, the protein sequence space is combinatorial. And in particular, if you want to sort of generate proteins, it's very expensive. And so we have to use guidance to kind of narrow down the combinatorial large space. Now, classifier guidance is great because you can have this sort of narrowing, to have this controllable generation with no additional training. As we know, we only have up to the evening, so training a diffusion model will just take too much time and it's kind of finicky. So we can actually make this tractable for this evening. The basic idea is based on this equation. As you know, there's a connection between diffusion models and score models. And essentially, it's learning the distribution, or you could say the gradient, the log probability distribution of the data. But classifier guidance is essentially adding this kind of additional term where you take the gradient of the thing you care about, so this kind of classifier. But the question is, what happens when you operate in the regime where the classifier is non-differentiable? For example, let's say you care about binding affinity, and the score that you get is by running a program, right? Some kind of docking software. Clearly that's not differentiable, right? You take a sequence and you upload it into the program, you run the program. And so the naive idea is, why not just do search in the logic space of the diffusion model? So actually this comes from, the intuition comes from this paper called Universal Guidance for Diffusion Models. This is actually from 2020, and this is on the image space, continuous space, much easier. But what they do is essentially take sort of this kind of local search around the hidden logics that generates the thing you want, right, on your diffusion model. And you can see this perturbation, and it's actually an optimization equation that they do. They do stochastic gradient descent. Whereas if it's not differentiable, then we can't do stochastic gradient descent, but we could just, for example, just do kind of naive search. For example, take your logits and do some kind of kind of random but diverse sort of perturbation, right? So I guess better than stochastic. That's the naive idea, and I'm wondering if it will work. So I guess how do we accomplish this in just a few hours by this evening? The first is we don't have to train a diffusion model. That's the great thing about classifier guidance, no additional training. The second is we'd have to go and just integrate some kind of guidance. So maybe it might be from docking software, it could be some value function that's trained online. And the last thing is just run some experiments and evaluate. And the great thing is it's not too hard because we're not training any models. Thank you.
Thank you, Justin.
Hey everybody, my name's Elliot. I run a community biotech lab called Celsius. I have also done biomanufacturing in a number of different ways. Let's see if I can plug this in. And, all right, if it doesn't show, then no worry. Oh. It's just a single slide, my bad. So I'm about to show a molecule that is responsible for the deaths of thousands of children every single year. This molecule is known as RDX, Research Development Explosive. It's found in most of the explosives that are used around the world. It is a very simple small molecule, and part of the thing that happens is that it occurs in landmines, and landmines cover the entire surface area the size of Wyoming. They kill up to 5,000, mostly children, every single year, and they have no shelf life, meaning that as landmines cover more and more of the Earth's surface, it makes it so that you can't actually go in those areas and actively interact in those places, but kids don't know that, and so they play in fields and get blown up. So this small molecule is something that could potentially be interacted with an enzymatic target, and thus degrading landmines and adding a shelf life to landmines themselves. Using protein diffusion models, as well as a number of other new metabolic engineering techniques, the idea is to generate a number of viable candidates of enzymes that could potentially degrade RDX, therefore adding a shelf life to landmines, putting it in soil bacteria, and giving people around the world the ability to open up land access and prevent kids from dying. Thank you.
Thank you, Elliot. Eric, you on reporting experiments from video.
No. Hey everyone, I'm Eric. What I'm thinking of doing is not exactly biology, rather it's the lab. So I spent 5 years working at Benchling, a software platform for life science, and mostly on our notebook to try to make it easier for scientists to plan their experiments and record the results of their experiments. So with all this like AI stuff so hyped, I was wondering what's new that we could do to make it easier for scientists to both plan and do their experiments, and possibly even make it easier to automate an experiment. And after doing some research, I realized one big blind spot right now is just what is not the biology itself, but just navigating a lab, like all the real world details of like, oh, if I want to like just like do like a PCR or something automatically, like I don't like have a robot that can like walk around and like take the right like sample from the freezer or like find the machine or anything like that. So just those very like basic real world tasks. Right now there's nothing. And so I remember this journal of visualized experiments, just like thousands and thousands of experiments of like video tutorials. And I was thinking, this is a very interesting evaluation data set for any kind of AI system that purports to understand anything about actually doing something in a lab, like whether it be like samples or reagents or equipment or actions or anything like that. I think a really neat hackathon-sized project is just to evaluate kind of the baseline of what state-of-the-art AI systems can do with these kinds of videos. So can common equipment be recognized? Can like Gemini know like if someone is pipetting, right? Just those very, very basic things. Is that even possible? So that's, yeah, that's what I want to do.
That is Eric on reporting experiments from video. Manu will discuss expressing opsins in specific neuronal cell types. Let's hear it for Manu.
I think if the HTML is not working, it's fine. All right. So I think I made a bunch of slides to discuss like some of the problem context. I think luckily the speakers before me have already explained it. I am a PhD student at MIT. I'm also an intern at Future House. I typically work on AI tools for protein engineering. And I think people are aware with AlphaFold and ESM3, there's a lot of different models that model protein structure and sequence, kind of in isolation. They don't ever consider like cellular context in which those proteins function, which what it leads to is you make libraries of proteins that you want to test for any specific function, and most of them don't work in the lab. So what I'm trying to do is figure out if there's a way that they can kind of do protein design with the cellular context. So the specific problem I'm trying to do is if you have a channel rhodopsin, could you, which is a protein that responds to light and is used in optogenetics as a way to study the brain, can you combine that information with expression profiles and regulatory DNA sequences to design new synthetic transcription factors and the promoter sequences? So the channel rhodopsin will only express in the neuronal cell type of interest. We collected a lot of data in relation to it. We collected gene expression data of mice genome and its expression across multiple different cell types. We collected all the transcription factor atlas of mice, and we also collected all the regulatory DNA sequences. So if anyone wants to work with us, that would be great. I'd be around.
Karen Sachs, the founder of NextGen Analytics, will talk to us about deep models for cancer therapeutics, ML for AML, and longevity.
It's on the spreadsheet, or as you like. Doesn't matter.
Do try to keep it 2 minutes. So.
Yeah, it'll be quick. It's not all those slides, Jeremy, don't worry. Okay, hi, I'm a computational biologist. I've been working in cancer, neurodegeneration, and other areas for quite some time. I look at the biological circuitry, so it's a natural fit for target discovery. And in the context of deep learning, the impetus in the space has been to take some patient-specific data and train it on perturbational data in biological cells and kind of do a black box approach. Oops. Jeremy, something went dead. Okay, and do a black box approach and predict what is the drug target. So if you have a cancer state and the cells are proliferating when they shouldn't be, if you can find the right thing to target in the cell to switch the pathway to one that's not proliferating incessantly, then you might cure the cancer. My background is in causal AI. I've been using I've been learning causal circuitry in biology using causal methods for many, many years. Recently, out of Stanford, there was, can we go beyond the black box models? So recently out of Stanford, a guy called Yusuf Rouhani published this beautiful model that started to take a step towards using causal methods in the context and bringing these black box models more towards a causal AI. He did this by capturing some of the causality in the embedding itself. I thought this was a really beautiful model. My proposal for today, and we worked on this actually at the last hackathon here, I think it was in April, and we actually put a little team together after that and have been working on this a bit since then, so it's really cool. Eric is here in the back, also has been working on this project. Uh-oh, it's dead again. Oh, so it's still here. So the idea is to take this really cool model from Rouhani and train it on patient-specific data using transfer learning and capture some of the causality on the underlying biological circuits, and then personalize a target for individual patients with the cancer AML. Now we're using AML because I have a little bit of expertise in AML. It's a leukemia cancer. But this could be expandable to anything. And in the context of the company that we kind of put together when we were here, we've been looking also at longevity applications, for example. So for example, how to reduce inflammation in a particular person's circuitry using their own personal molecular data. Yeah, so come talk to me if you're interested in any of this. Thank you.
Hello, I just had this thought while I was here. Basically, I do some fine-tuning DPO of some protein models on the side, and I was talking to my friend who's like, that's going to be a security concern in the future in terms of like linking up with that, going for it. So I'm thinking of almost like a Hugging Face-esque interface for academic labs that's super secure and super certified to allow them to do that, to kind of cut that off in the future. But that was just the thought I had while going through this. That might be a need in the future in terms of security, safety, and regulation coming up. Thank you.
Very cool. And Gary on XR for reproducible science.
Hi guys, I'm Gary. Matthew and I are taking a stab at the reproducibility questions in science. And we're looking to provide scientists a new way to instruct one another how to do their experiments. So via LLMs and AI, we're able to break up your published papers and protocols into actionable steps. And some actionable steps we can actually guide through XR. So you'll put a Vision Pro or a better XR device on in the future, and we'll guide you through those steps. And imagine animal experiments, you can actually literally tell people how to insert a capsule and cut through the skin. And we'll check if you do things correctly. If you do things incorrectly, we'll evaluate if it's catastrophic or it's fixable, and even suggest how to fix those issues. The person publishing those papers can actually customize how to fix issues with us, so it becomes a new way to instruct one another. So we're actually trying to do what Journal of Visualization does, but better. So happy to chat with all the immense research expertise in the room. Thank you.
Okay, fantastic. So it seems that we have a number of nascent project ideas. There's a brilliant conversation, I think, to be had in the opener about what exactly everyone wants to build today, but I can't wait to see what you guys are going to create. Just a few logistics. So we have lunch for everyone. I'm sure you're hungry. So Chinese food is set up in the back for you all. This is a 4-story mansion, so feel free to take the floor that we're on, the floor below it, and the floor below that as open hacking space. So do find a cozy corner, make yourself at home with your team, and build something for the next few hours. So it's about 2 o'clock, so we have 6 hours of hacking until demos at 8 o'clock. So do get to find a really great team. It's a fantastic opportunity to come up with something creative, build something, show it off. As you just heard from Karen, it's possible that the team that you build something of interest with will meet with you over and over again on a new project of interest. So do take full advantage of the opportunity. And with that, let the AI for Biological Sciences Hackathon begin.
Krishna, that was a really cool presentation.
Thank you so
Nice
much.
to meet you.
Yeah.
So you just did this over the weekend?
Yes, yeah. No, the last 2 days.
I actually have the same question. Did you have to have a cluster to turn from?
We don't have a cluster.